All right, now let's start our discussion over the cardiovascular system, and this is going to be the first lecture in a three-part series. We are going to talk about blood first, and then we will move on to the heart, and then we will finish with a discussion of blood vessels. So let's go ahead and get started first here with blood. All right, so what is blood? Well, you may recall from A&P 1 that blood is a specialized form of connective tissue, of fluid connective tissue, and the reason for this is because it's going to be made up of cells, such as red blood cells and white blood cells, but there's also going to be an extensive extracellular matrix, and that is the plasma portion of the blood. So blood is going to be transported through the cardiovascular system. It's going to be pumped by the heart. It's going to be carried away from the heart by arteries. It's going to diffuse into the tissues, allow gas exchange between blood and body tissues at the capillary level. And then it's going to come back to the heart through vessels called veins. All right, what is blood composed of? Well, like I said, it's going to be composed of cells and then an extracellular matrix. So the cells are the formed elements of the blood. Okay, so this is going to include things like erythrocytes, leukocytes, and platelets. Erythrocytes are red blood cells. These guys transport respiratory gases such as oxygen and carbon dioxide through the blood. Erythrocytes are better called formed elements because mature erythrocytes lose their nucleus and their organelles. So they're not really functioning like a cell normally functions. They essentially become sacs of hemoglobin, and we'll talk about that here in a minute. Next in your formed elements, you have leukocytes. These are the white blood cells, and these guys defend us against pathogen invaders, okay? So against things like bacteria and viruses. Platelets. These guys help to form blood clots to prevent blood loss, and they're formed out of a larger cell that basically breaks up into these little pieces that we call platelets. So again, platelets are better referred to as a formed element. And then you have the extracellular matrix, or the plasma of the blood connective tissue. Plasma, again, is going to be the fluid portion of blood, so that's going to be water, plasma proteins, and dissolved solutes. The functions of blood include transportation, protection, and regulation of certain body conditions. Okay, so transportation, blood is going to transport formed elements, so the red blood cells, the white blood cells, the platelets, as well as those dissolved molecules and ions that are dissolved in the plasma. So those formed elements, the red blood cells, are going to carry oxygen to the tissues and carbon dioxide from the tissues back to the lungs. And blood is going to transport nutrients that we absorb from eating. It's going to transport hormones throughout the blood, as well as heat and waste products. Protection. This is going to be mainly the role of the leukocytes, but there are also plasma proteins and some other molecules of the immune system that are transported throughout the blood that protect us against pathogens. And then platelets and some plasma proteins are going to protect us against blood loss, and we'll talk about that in more detail at the end of this lecture. All right, what body conditions does blood help regulate? Well, body temperature, body pH, and fluid balance. So blood absorbs heat from the body cells, especially our muscles, and then it transports that heat through our body, and it's going to be released at the blood vessels of the skin. Our pH, so blood is going to absorb acid and base from our body cells, and the blood itself contains these things called buffers, which help keep the pH of the blood within a very specific narrow range. And it's very important to keep the pH in that narrow range because our enzymes only function in a very narrow range of pH. Finally, blood helps to control fluid balance. So water is going to be added to blood from our GI tract, and then water is going to be lost through urine, through our skin, and even through respiration. All right, some physical characteristics of blood. So the color of blood is going to depend on how much it's oxygenated. So oxygen-rich blood is going to be bright red, while oxygen-poor blood is dark red. As far as how much blood we have in our body, we have about 4 to 6 liters, depending on the person. 5 liters is a good estimate for the average adult. Viscosity. Blood is going to be 4 to 5 times thicker than water, and this is because it has those formed elements and dissolved substances in it. So this viscosity is going to depend on the amount of dissolved and suspended substances relative to the amount of fluid. If your red blood cell number increases, your viscosity is going to increase. If the fluid portion of your blood decreases, viscosity is going to increase. Blood is going to exert osmotic pressure, okay? So it has in its plasma a bunch of proteins and ions dissolved. And so the concentration of these proteins and ions is higher inside of the vessel, the blood vessel where the blood is, than outside of the blood vessel, like in the tissues where interstitial fluid is. So what happens is blood essentially draws that interstitial fluid in to the blood vessels because of this osmotic pressure that blood is exerting. We said blood helps regulate heat, so blood is about 1 degree Celsius higher than the rest of the body, and so it's going to warm the areas through which it travels. Blood pH, we said, needs to be maintained in a very narrow range. Blood is slightly alkaline or slightly basic, so pH is between 7.35 and 7.45. Remember, 7 is neutral, and anything above 7 is going to be basic. Anything below 7 is going to be acidic. Now, this slightly alkaline range of 7.35 to 7.45 is crucial for plasma protein shape. Okay, and a lot of these plasma proteins are enzymes, and so they really need their shape in order to function correctly. And if you mess with that shape, you can denature those proteins, and then they lose their shape permanently, and they're not functional. All right, a little bit more on the components of blood. So what they can do is they can take whole blood and put it in a tube, and put that tube in a centrifuge. And the centrifuge basically spins that blood down, and as it spins it down, it separates it into different layers depending on the density of each one of those layers. The bottommost layer, the densest layer, is going to be composed of erythrocytes, okay? And this makes up about 44% of whole blood. There's going to be a tiny middle layer called the buffy coat. This is about 1% of whole blood, and it's going to be composed of leukocytes and platelets. And then the very top layer is plasma. This is straw-colored liquid, and it makes up about 55% of the plasma of the sample. All right, so here's that tube of whole blood. They drew it from the patient, took that tube, put it in a centrifuge, and it spins it around. And the densest stuff is going to layer at the bottom, while the less dense stuff is going to be on the top. So let's look at this top layer first. This is the plasma, and plasma is going to be made up mostly of water, and then proteins such as albumins, globulins. This will include things like your immunoglobulins, like antibodies, fibrinogen, which is involved in the blood clotting process, and then other solutes as well, so electrolytes, nutrients, respiratory gases, so O2 and CO2, and waste products. Then you look at this very thin middle layer, the buffy coat, and that's going to be composed of platelets and the various different white blood cells, which we'll talk about here shortly. And then finally the bottom layer, which is composed of red blood cells. And remember, this bottom layer makes up about 44% of whole blood. And that bottom layer, composed of the red blood cells, is also known as the hematocrit. So what the hematocrit is just the percentage of whole blood that is red blood cells. Now, hematocrit can range from about 38% to 56%, depending on the person. Those who produce testosterone are going to have a higher hematocrit than those who don't, because testosterone causes erythropoietin to be secreted by the kidney. Remember, erythropoietin is a hormone we'll talk about here shortly, a hormone that essentially signals the red bone marrow to make more red blood cells. All right, and you may have heard this term before, blood smear. What is a blood smear? Well, all that is when you take blood and you place it on a microscope slide, stain it so you can see the cells, and look at those cells underneath a microscope. The different formed elements are going to differ in how they appear. So your red blood cells are going to be the most numerous out of all the formed elements. They're going to appear pink. They're not going to have a nucleus, and they're these biconcave discs. Leukocytes are going to be larger. They vary in their form. They have very noticeable nucleus. And then you have platelets. These are small fragments of cells. All right, so looking at a blood smear, you can see these pink guys here are red blood cells, and they're very numerous. Then you see these larger cells that are kind of stained whitish purplish. These are going to be your leukocytes, and there's various different forms of leukocytes. For example, this is a neutrophil, this is a monocyte, and this is a lymphocyte. We'll talk about all of those in detail. And then you can see tiny little fragments of cells here, and these are the platelets. All right, plasma, we mentioned, is mostly composed of water. And then it's got a bunch of different plasma proteins in it, and dissolved molecules and ions and gases. It's similar in composition to interstitial fluid, so that's going to be the fluid that surrounds the cells in our tissues and organs, okay? But plasma has a higher protein concentration than interstitial fluid, and you'll see this has the effect of being able to draw interstitial fluid into the blood vessels. All right, blood is a colloid, and essentially what a colloid is it's a mixture of microscopic particles that are suspended in a fluid, and these microscopic particles do not settle out of the suspension. Okay, so that's what a colloid is, and in blood it's going to be the dispersed plasma proteins that are suspended. These plasma proteins include things like albumin, globulins, fibrinogen, and the other clotting proteins, enzymes, hormones, and most of these guys are going to be produced in the liver. However, some are produced by other organs or even white blood cells. Okay, and those plasma proteins exert colloid osmotic pressure. Okay, and all that means is that there's a higher concentration of those proteins inside of the blood than outside of the blood. So through osmosis, blood is going to draw fluid from the interstitial spaces into the capillaries, okay, and that's going to help mitigate fluid loss as gases are exchanged in the tissues. And we'll talk about that more in detail a little bit later. Now, there are some diseases that mess with the colloid osmotic pressure. For example, some liver diseases are going to decrease the liver's ability to produce those plasma proteins. So there's fewer plasma proteins circulating in the blood, there's less colloid osmotic pressure, so more fluid can be lost in the tissues, okay? And that can result in tissue swelling. Kidney diseases. There are several kidney diseases that increase the elimination of plasma proteins. So again, you're going to have fewer plasma proteins circulating in the blood, less colloid osmotic pressure exerted, and more fluid is able to escape the bloodstream into those interstitial tissues, interstitial spaces. Okay, what are the plasma proteins? Well, we mentioned there's albumins. These guys are the most abundant group of the plasma proteins, and so they exert the greatest colloid osmotic pressure. And they act as transport proteins for some lipids, hormones, and ions. Then you have the globulins. This is the second largest group of plasma proteins. You have alpha globulins and beta globulins, and these guys are also transporters. And then you have the gamma globulins, or we also call these immunoglobulins or antibodies. And these guys are part of the body's defenses. Next in the plasma proteins is fibrinogen. This makes up only about 4% of plasma proteins, but it's crucial in blood clot formation. Now, fibrinogen is soluble in water. That is, it can be dissolved in water. But following trauma, it's going to be converted to insoluble fibrin. And it's this fibrin that helps form the blood clot itself. You may have heard the term serum before. Serum and plasma are not the same thing, okay? All serum is plasma once the clotting proteins have been used up. And then you have the regulatory proteins that are circulating in plasma, and this is going to make up less than 1% of total plasma proteins. It's going to include things like enzymes and proenzymes. Now remember, enzymes catalyze biological reactions. That is, they make the reactions go faster than they normally would. And proenzymes are enzymes that need to be activated before they become an enzyme, okay? Okay, in addition to being a colloid, blood is also considered a solution. And a solution is going to be a liquid that has substances completely dissolved in it. And these substances in the blood are going to include electrolytes, nutrients, gases, and waste products. Okay, polar or charged molecules are going to dissolve easily in plasma, and that's because polar and charged substances are hydrophilic. That is, they love water, so they can interact with the water of the plasma. Nonpolar molecules, on the other hand, they don't get along with polar substances. So a nonpolar molecule is going to require some sort of carrier protein in order to help carry it through that water and dissolve it in the water. All right, hematopoiesis. This is the production of formed elements. So those red blood cells, the white blood cells, and the platelets. So it's going to occur in the red bone marrow of certain bones. And it's going to involve stem cells. And these stem cells are called hemocytoblasts. Hemocytoblasts are pluripotent, and that just means they can differentiate into many different types of cells. Okay, so as far as stem cells go, you can have pluripotent, which, pluri comes from plural, meaning more than one, potent meaning potential, so it has the potential of becoming more than one type of cell. And then you can have omnipotent. That means all potential. Omnipotent stem cells are typically what you see in the developing embryo. They can become any type of body cell. But in hematopoiesis, we are working with pluripotent stem cells. Now, pluripotent stem cells can produce two different cell lines. There is the myeloid cell line and the lymphoid cell line. The myeloid cell line forms the red blood cells, all the white blood cells except for your lymphocytes, and it forms the megakaryocytes, which produce platelets. The lymphoid line only forms lymphocytes. Then there's these substances called colony stimulating factors. And what these guys do is they signal the red bone marrow to begin hematopoiesis. Okay, here's kind of an overview of hematopoiesis. So you can see we start up here at the top with a hemocytoblast. This is a pluripotent stem cell. And this guy can eventually become any one of these guys down here, an erythrocyte, platelets, white blood cells. Okay, so that's what we mean when we say pluripotent. Now, this guy can, depending on the signal it receives, go on to become a myeloid stem cell or a lymphoid stem cell. Let's say it goes on to become a myeloid stem cell. Well, this myeloid stem cell is going to develop into a red blood cell, platelets, or a type of white blood cell based on the colony stimulating factor signals that it receives. So let's say this myeloid stem cell receives signals involving multi-CSF and EPO. It's going to go down this pathway of erythropoiesis. You have a progenitor cell that differentiates into a proerythroblast, then differentiates into an early erythroblast, then it becomes a normoblast. At this point, the red blood cell is going to lose its nucleus. Then it's called a reticulocyte, and a reticulocyte is almost a red blood cell. It just still has some organelles. So the reticulocyte is going to lose its organelles, and then it's what is known as an erythrocyte. And remember, an erythrocyte is basically just a sac or a bag of hemoglobin, and hemoglobin transports oxygen as well as carbon dioxide. On the other hand, this myeloid stem cell could have received signals to start undergoing thrombopoiesis, and this is going to be how platelets are made. So you start again with a progenitor cell. It becomes a megakaryoblast. Under thrombopoietin, it becomes a pro-megakaryocyte, and finally a megakaryocyte. And what the megakaryocyte does is it has these extensions of the plasma membrane that it sticks out into the blood vessel, and these pieces get sheared off from the main cell, and it's those pieces that are the platelets. Okay, or the myeloid stem cell could undergo different signaling to become a different type of leukocyte, so either one of the granulocytes or a monocyte. We'll talk about these guys in detail here shortly and what they do. Now, going back up here to the hemocytoblast, let's say that the hemocytoblast, the pluripotent stem cell here, received a signal to undergo the lymphoid line differentiation. And then that lymphoid stem cell then can either become a natural killer cell, a T cell, or a B cell, depending on the signals that it receives. Okay, here's the steps of erythropoiesis. This process requires iron, B vitamins, and amino acids. And this is going to be important when we get to causes of anemia, okay? Big cause of anemia can be iron deficiency, as well as deficiency in some B vitamins. Remember, red blood cells are going to come from the myeloid stem cell, the myeloid line, and it's going to respond to different colony-stimulating factors until it becomes a reticulocyte. And a reticulocyte is going to lack organelles except for ribosomes, and then that reticulocyte is going to lose its ribosomes and finally become an erythrocyte. All right, leukopoiesis. This is just the fancy term for the production of leukocytes. Remember, this is your white blood cells. And your white blood cells are going to include the granulocytes, the monocytes, and the lymphocytes. The granulocytes are the neutrophils, the basophils, and the eosinophils. Remember, these guys are going to derive from the myeloid cell line. Also arising from the myeloid cell line are the monocytes. And monocytes, what they're going to do is they are eventually going to leave the bloodstream and differentiate into macrophages. And macrophages phagocytose things, okay? They engulf pathogens and debris and break them down inside of their cells. Okay, and then finally, remember you have the lymphocytes, and these guys are not myeloid cell line, they are lymphoid cell line. You've got lymphoid stem cells that are going to differentiate into these things called lymphoblasts, and then the lymphoblasts mature into the actual lymphocytes. Okay, so you're going to have B lymphocytes and T lymphocytes. Alternatively, some lymphoid cells can differentiate into natural killer cells. And natural killer cells basically patrol the body for infected or cancerous cells and destroy them. Your B cells and your T cells are going to be involved in adaptive immunity. And we'll talk about this more in detail when we get to that chapter. But just, in a nutshell, The T cells are going to target cells that are infected. The B cells are going to produce antibodies that act on pathogens and infected cells. All right, and then you've got thrombopoiesis, and this is platelet production. Remember, platelets are going to arise from the myeloid stem cell line. Under the influence of thrombopoietin, a megakaryoblast becomes a megakaryocyte. And that megakaryocyte produces thousands of platelets. Like I said, what they do is they produce these long extensions of the plasma membrane called proplatelets, okay? And these extensions extend through the blood vessel wall, and then the force of the blood rushing by basically shears off these pieces of these proplatelets, and that's what become the platelets. Here's a better image of those proplatelets. You can see them extending through the blood vessel wall, and it's the blood flow itself that's going to basically slice off pieces of those proplatelets, and then at that point they are called platelets. All right, now we're going to go into detail on each one of those formed elements. So we'll talk extensively about the erythrocytes, or the red blood cells. Then we'll move on to the leukocytes, and then finally we'll finish up with the platelets. So the erythrocytes, we said these guys lack a nucleus, they lack organelles. They are packed full with hemoglobin. They have a biconcave disc structure, and they're small and flexible. What they do is they transport oxygen and carbon dioxide between the tissues and the lungs. Okay, so they're going to pick up oxygen as they pass through the lungs and give off carbon dioxide in the lungs. Then they're going to go to the tissues where they give off oxygen and pick up carbon dioxide. And then they go back to the lungs and the cycle begins over again. Okay, so here's what they look like. If you cut one kind of down the middle here, you can see it's a biconcave disc, so it caves in from the top and it caves in from the bottom. And there's no nucleus, no organelles, just hemoglobin, chock full of hemoglobin. And this is what they look like in a blood vessel under a light microscope. Okay, so we said erythrocytes are packed full with hemoglobin. Well, what exactly is hemoglobin? It's going to be a red pigmented protein that transports oxygen and carbon dioxide. When it is maximally loaded with oxygen, we call it oxygenated hemoglobin. When it has lost at least a little bit of oxygen, we call it deoxygenated hemoglobin. Okay, each hemoglobin molecule is going to be composed of 4 globins. Okay, and that's going to be 2 alpha chains and 2 beta chains. And all that means is that the alpha chains are proteins that are identical to one another, and the beta chains are proteins that are identical to one another. Okay, so each one of those chains is going to have a heme group. And all a heme group is this ring structure called a porphyrin with an iron ion right in the middle. And what happens is oxygen binds to that iron ion. Okay, so if each one of these chains has a heme group, and each one of those heme groups can bind an oxygen molecule, then each hemoglobin can bind up to 4 oxygen molecules. Okay, so here is the hemoglobin molecule itself. You can see it's composed of 2 alpha chains, it's these dark brown proteins here, and 2 beta chains, these tan proteins. And each one of those chains has a heme group inside of it. So if we blew up that heme group, we would see a ring structure here. This is the porphyrin ring. And in the middle, an iron ion. Iron is designated as Fe on the periodic table. And what is oxygen binding to specifically? Remember, it's binding to this iron ion in the middle of the heme group. So each hemoglobin molecule has 1, 2, 3, 4 hemes, so it can bind up to 4 oxygen atoms. Okay, we said oxygen is going to bind to iron. Now the binding is relatively weak, and this is important because you want hemoglobin to give off its oxygen, release its oxygen when it gets to the tissues. So since this bond is weak, that can happen. Carbon dioxide is going to bind to the globin protein, not to the iron ion itself. And again, you're going to see binding is relatively weak, so this is going to allow for that carbon dioxide to detach in the lungs. All right, erythropoietin, a little bit more about it. It's also called EPO, and remember, it controls erythropoiesis, or the development of red blood cells. EPO is primarily produced in the kidneys. There's a little bit that's made in the liver, and it's going to be secreted in response to a decrease in blood oxygen levels. So what happens is you're going to have a drop in your blood oxygen levels, and then those cells in the kidneys and in the liver are going to recognize that, and they're going to begin secreting EPO. EPO is going to travel to the red bone marrow, where it's going to act on those myeloid cells. Remember, erythrocytes come from the myeloid cell line. And in response to EPO, those myeloid cells are going to differentiate into erythrocytes. So the overall picture is you get more red blood cells. Why is that important? Well, because you're going to have more red blood cells, you're going to have more hemoglobin in the blood, and so more oxygen carrying capacity in the blood. So that's going to increase your blood oxygen levels, and when that happens, you're going to have a negative feedback mechanism, and that increase in blood oxygen levels is going to tell the kidneys and the liver to stop secreting EPO. What stimulates EPO? Well, blood oxygen levels we mentioned, but also testosterone and some environmental factors like altitude. So testosterone is going to stimulate EPO production in the kidney, and this is why you're going to see men and people on testosterone therapy have a higher erythrocyte count and a higher hematocrit. Some environmental factors can influence EPO levels. For example, low oxygen levels at high altitude is going to stimulate EPO production, and that's going to stimulate an increase in red blood cells. And here it is laid out in a graphic for you. So you're going to have the stimulus, remember, is going to be decreased blood oxygen levels. The receptor is in the kidney, and the control center is in the kidney, and so the receptor detects that decreased blood oxygen, and those specialized cells in the kidney release EPO into the bloodstream. EPO is going to travel through the bloodstream until it gets to the bone marrow, where it's going to act on the myeloid stem cells. And that's going to make a bunch of those myeloid stem cells become erythrocytes. So you're going to get an increase in red blood cells throughout the blood, and therefore an increase in hemoglobin and an increase in oxygen carrying capacity. That is going to increase blood oxygen levels. The kidney senses that increase in blood oxygen levels and tells its cells to stop secreting EPO. And remember, that's a negative feedback mechanism. Okay, a little bit about blood doping. What is blood doping? Well, that's where you increase the number of red blood cells that you have so that you can carry more oxygen and, in theory, perform better as an athlete. There's a couple of different methods that are used in order to do blood doping. One method is self-donation of red blood cells. And this has a two-pronged effect. Okay, so what the person is going to do is they get a pint or two removed from their blood, and then that's going to actually lower their oxygen carrying capacity, and that's going to stimulate those kidney cells to start secreting EPO. Okay, so now they're making red blood cells again. However, right before competition, they go and transfuse that blood that they removed from themselves back into their bloodstream. So not only are they making EPO kind of at a higher level than they normally would at homeostasis. Now they've just given themselves transplanted red blood cells straight into their bloodstream. So they're going to have a really high oxygen carrying capacity. Another method of blood doping is just to use pharmaceutical EPO. Just stimulate your bone marrow to keep churning out red blood cells. Now, blood doping is a terrible practice, and it's a terrible idea. Why? Well, you're artificially increasing your blood viscosity. So this is going to increase risks for things like stroke. The heart is going to have to work harder because you have more viscous blood, right? And that can damage the heart. In fact, you can have permanent cardiovascular damage from blood doping. And of course, for reasons, in addition to physical safety, but also ethical reasons, blood doping is banned in athletic competition. All right, erythrocyte destruction. How and why are red blood cells destroyed? Well, remember, they lack a nucleus and organelles, so they're not going to be able to make proteins to repair any damage that they incur. So they have a limited lifespan of about 120 days. What happens is old blood cells, red blood cells that are traveling through the spleen or through the liver, get phagocytosed by macrophages, okay? And those macrophages break down the globin and membrane proteins into amino acids. Your body's going to use those amino acids to make more proteins eventually. And then that iron from the hemoglobin is going to be transported to the liver. It's transported to the liver by a carrier molecule called transferrin. And then when it gets to the liver, it's going to be bound to storage proteins or storage molecules, ferritin and hemosiderin. And basically, the liver and the spleen act as your iron storage areas, and then as needed, they will transport iron to the red bone marrow as red blood cells are being made. Okay, so that's how the globins and the membrane proteins are broken down, and that's how the iron is recycled. Now let's talk about what happens to the heme group. Okay, so this is just that porphyrin ring without the iron, because the iron has already gone to the liver and the spleen. So what happens is macrophages convert that heme group, that porphyrin ring, into a green pigment called biliverdin. Biliverdin then gets converted into bilirubin, which has a yellow pigment. At this point, it is unconjugated bilirubin, and so it's going to be transported to the liver by albumin. The liver converts it to conjugated bilirubin, and it's conjugated bilirubin that becomes part of the bile, and bile is used in digestion. Bilirubin is then going to be converted into urobilinogen in the small intestine. And then it can go one of two ways. It can either continue through the intestine, and the bacteria are going to convert it to stercobilin, and that is expelled from the body as the brown pigment in feces, or urobilinogen can be absorbed back into the blood, converted to urobilin, and be excreted from the kidneys, and that's what gives the yellow pigment to urine. Okay, why do some people get jaundice? This is a condition where the skin yellows, the whites of the eyes yellow, okay? And you see this in people who have liver disease. And one of the reasons for this is because that unconjugated bilirubin can't be taken up by the liver. Okay, so it just hangs out in the bloodstream and it gets deposited in the tissues. And that's what jaundice is. Okay, anemia. What is anemia? Well, a lot of people think anemia just means you have low iron or low red blood cells. But really what it is it's a condition where the blood has a reduced ability to carry oxygen. So this could be due to fewer red blood cells than normal, but it could also be due to a deficiency in hemoglobin or abnormalities in hemoglobin that prevent it from being able to carry oxygen normally. So if you can't carry oxygen normally, you can't get it to your tissues where it's needed so that your tissues can make ATP, right? So you're going to have symptoms of lethargy. You're tired, you're fatigued, you don't have enough energy because you're not getting oxygen to the tissues where energy can be made. You can also get shortness of breath, pallor, which is a pale skin tone, and palpitations. There are many types of anemia. Here are just a few. So there's aplastic anemia. This is going to be some problem in the red bone marrow, okay? The red bone marrow cannot make the red blood cells like it should. And sometimes this is due to poisons. Some drugs can cause this, and radiation can even cause it. There's congenital hemolytic anemia. A hemolytic anemia is any anemia where the red blood cells are being lysed or broken down, broken apart. Okay, so hemolytic. Again, the erythrocytes here are going to be destroyed, and this is due to a genetic defect. Then there's erythroblastic anemia. This is going to involve large numbers of erythroblasts, which are those immature red blood cells, right? And the reason for this is because the cell maturation has been accelerated. So the red blood cells are dividing too soon and getting released too early. There's hemorrhagic anemia. This is going to be due to blood loss. There's pernicious anemia. This is actually an autoimmune disorder. And what happens is the patient cannot make this thing called intrinsic factor. And intrinsic factor is necessary in order to absorb vitamin B12. Vitamin B12 is necessary in order to make mature red blood cells. So if you don't have vitamin B12, you cannot make red blood cells. And then, of course, there's sickle cell anemia or sickle cell disease, and this is due to a genetic defect in which the patient's hemoglobin is abnormally shaped. And what happens there is basically the hemoglobin, because it's abnormally shaped, it starts to clump up in the cells and creates kind of a sickle or a C-shaped cell. And those red blood cells do not move very well through the blood vessels, especially the smaller blood vessels, and they tend to get caught and break apart, and so these patients tend to have lower numbers of red blood cells than normal. Treatment of anemia is just going to depend on the underlying cause of the anemia. For example, if you have iron deficiency anemia, treatment might include iron infusions. If you have anemia due to acute blood loss, they're probably going to give you blood to replace that, okay, and to prevent hemorrhagic anemia. If you have anemia due to, say, kidney disease, and so your kidneys can't produce EPO, the treatment there might be pharmaceutical EPO. Okay, so treatment is varied, and it really just depends on the underlying mechanism behind the anemic state. Okay, let's move on to blood types. So a person's blood group is going to depend on surface antigens that project from the membrane of those red blood cells. Okay, so let's talk about the ABO blood group first. So you've heard of people who are type O, type B, type AB, etc. That's determined by whether or not they have an A antigen and a B antigen on their red blood cells. So in type A blood, these people are going to have red blood cells that have the surface antigen A only. In type B blood, that's going to involve red blood cells that have only the B antigen. Then there's type AB blood. These people have red blood cells that have both the A and the B antigens. And then if you're like me and you're type O blood, your red blood cells have neither the A or the B antigens. Okay, your body makes antibodies against some of these antigens, and that which antigen it is depends on which ABO blood type you are. So, a little bit about those antibodies. There's anti-A antibodies and anti-B antibodies. The anti-A antibodies, as you would expect, react with antigen A. The anti-B antibodies react with antigen B. Now, it should make sense that you do not produce antibodies for your own surface antigens. That would be a huge problem. Your immune system would essentially recognize your red blood cells as foreign and destroy them. What you do have are antibodies to antigens that are foreign to you. So a person with type A blood has the A antigen on their red blood cells, and they produce anti-B antibodies. A person with type B blood has type B antigen on the red blood cell surface and produces anti-A antibodies. If you are type AB, you are going to produce neither anti-A or anti-B antibodies. That should make sense because you have antigen A and antigen B on your cell surfaces. You don't want to destroy them with anti-A and anti-B antibodies. Finally, if you're like me and you're type O, you have neither antigen, but you make antibodies to both A and B antigens. So you have both types of antibodies circulating in your plasma. All right, so here it is illustrated for you. Here's type A. Here's what the red blood cells of a person with type A blood would look like. And these things projecting off of the cell surface are antigen A. And this is what their plasma would look like. They would have anti-B antibodies circulating in their plasma. A type B person would have just the opposite. They would have red blood cells with surface antigen B and anti-A antibodies circulating. Type AB is going to have both antigens and neither antibodies. Type O is going to have neither antigens but both antibodies. And I want you to be intimately familiar with this and understand the logic behind why someone with antigen A will not produce anti-A antibodies, okay? So really study this chart. Make sure you know these inside and out. All right, so that was ABO. Now let's talk about the RH factor. This is also sometimes called surface antigen D. Whether you are positive or negative depends on whether or not you have the RH factor on the membranes of your red blood cells. I am O positive. So remember, I don't have any antigen A or antigen B, but because I'm positive, I do have the RH factor hanging off of my red blood cells. Now, in Rh negative people, they are not going to just produce anti-D antibodies. What they need to have happen first is they need to be exposed to someone with Rh positive blood. And so then their immune system will see that Rh factor and begin to build a response to it. And as mentioned before, the Rh type is indicated by positive or negative. So for a blood type to be given, you're going to start with the ABO group that they belong in, and plus or minus, they have that Rh factor. So AB positive person would have both A antigen and B antigen, as well as the Rh factor hanging off of their red blood cells. And this is just showing the Rh blood types. So if you're Rh positive, you have that surface antigen D or Rh factor on your cells. You're not going to produce antibodies to this. If you're Rh negative, you don't have that surface antigen D or Rh factor. But you're still not just going to automatically produce antibodies. What's going to happen is you're going to need to be exposed to Rh positive blood first. And this has some important implications when it comes to pregnancy. Okay, why do we care what our blood type is? Well, this is critically important when it comes to blood transfusion. So if you were to receive a transfusion from someone with an incompatible blood type to yours, what's going to happen is those red blood cells are going to enter your circulation, and your body is going to notice the antigens on them and recognize them as foreign. And then your body is going to send antibodies to those antigens, and it essentially clumps up all of those foreign red blood cells together. Now, why is that a problem? Well, if you've got clumps of red blood cells traveling through your vessels, they can block smaller blood vessels and prevent normal circulation. Additionally, those clots and blockages can cause hemolysis, rupture of the erythrocytes, and ultimately cause organ damage. Okay, now let's just look at this chart. There's a lot of information here, but what I want you to focus on is the universal donor, which is O negative, and the universal recipient, which is AB positive. So, O negative can give to any blood type there is. Why is that? Well, remember, type O lacks surface antigens A and B. So if it doesn't have those antigens, it's not going to trigger an immune response in your body to produce antibodies against it. It also lacks Rh factor. Again, your immune system cannot build a response to something that is not there. So O negative kind of just comes into the circulatory system and doesn't get recognized as foreign by your body, and so it's able to circulate normally. Okay, why is AB positive the universal recipient? Well, remember, AB positive is going to have all of those surface antigens: surface antigen A, surface antigen B, and the Rh factor. So it should make sense that they are not going to produce antibodies against their own cells, their own antigens. So what happens here is you've got a patient who doesn't have any antibodies to any of the blood groups, the ABO groups or the Rh group. So no circulating antibodies. They're not going to react to any transfused blood from any blood type. Okay, here's an example of erythrocyte agglutination. So what we have here is a person who's type B. They are receiving blood from a type A donor. So this is an incompatible blood match. What's going to happen is you can see the recipient's erythrocytes here, and then here are the donor's erythrocytes. But here are the recipient's anti-A antibodies, right? Because he's type B, he produces anti-A antibodies. And these anti-A antibodies are going to attach onto the type A red blood cells and cause them to clump up. And you can see here is one such clump, an agglutinated mess right here. And you can see it's blocking blood flow. So everything down this vessel past this clot is not getting oxygen, and that's called ischemia. And that damages tissues, it damages organs, and it can eventually lead to death. Death of the tissues, death of the organs, and death of the organism. All right, here's the agglutination test. So you can take a little bit of blood from the donor and a little bit of blood from the recipient and mix them together, and then look and see if they clump, and that's called an agglutination reaction. So here we start with type A blood of the donor. Here is the type A blood of the recipient, and you'll notice type A blood of donor has A antigens. the type A blood of the recipient has anti-B antibodies. So these anti-B antibodies are not going to interact with these A antigens at all. There's going to be no agglutination that happens, and you're not going to have a transfusion reaction or problem here. Okay, no clumping seen. If, on the other hand, you took type A blood of the donor and tried to give it to a person with type B blood, remember, a type B person produces anti-A antibodies. Well, those anti-A antibodies are going to interact with that surface antigen A and cause these cells to clump together. And you can see that that's what's happening right here. All right, remember we said that Rh positive patients are not going to produce anti-Rh antibodies, and that makes sense because they wouldn't want to produce antibodies that are going to interact with their own cells. However, recall that Rh negative people also do not just produce anti-Rh antibodies. What has to happen first is they need to be exposed to Rh positive blood. And how this typically occurs is through childbirth. So let's say you have an Rh negative mother, let's say she's O negative, and her baby is O positive. Well, during childbirth, it is not uncommon for a little bit of blood, baby's blood, to get into mom's bloodstream. And when this occurs, an immune response is going to happen. So mom's body is going to see that O positive, see that positive and go, that's foreign, let's produce antibodies against it. So now mom is going to have anti-Rh or anti-D antibodies. This is not a problem for the first pregnancy. The first pregnancy, baby does just fine. What it's a problem for is future pregnancies. So if mom, Rh negative mom, O negative mom, has another baby who is Rh positive, her antibodies may cross the placenta and destroy baby's red blood cells. And this is called hemolytic disease of the newborn. And what happens here is you get a baby who is typically severely anemic. They have hyperbilirubinemia, okay, so a lot of bilirubin in their system because all of those red blood cells are being broken down. And they can even get heart failure because they're not able to carry much oxygen in their blood. The heart, trying to compensate, pumps faster and harder, trying to get more and more blood to the tissues, and that wears the heart out, and the heart can go into failure. How do we prevent this? Well, in mom's second pregnancy, she's going to get a shot of RhoGAM. And what RhoGAM is basically special immunoglobulins. And what they do is they travel to baby's red blood cells and cover up those antigens, that Rh factor, on baby's red blood cells. Okay, so that was all red blood cells, erythrocytes. Let's move on to the leukocytes and talk about our white blood cells a little bit. So remember, white blood cells' primary role is going to be to defend against pathogens. And a pathogen is just, a microorganism that can make us sick. Now, your white blood cells, unlike the red blood cells, are going to contain a nucleus, and they're going to contain organelles, and they're going to lack hemoglobin. So they really have no role in oxygen carrying. These guys are motile and they're flexible. Most of them can be found in the tissues, okay? And what white blood cells do is they do this thing called diapedesis. And it's where they squeeze in between the endothelial cells of capillaries and basically squeeze through that blood vessel wall out into the tissues. And they do this because they are attracted to chemicals at, say, a site of infection. And that's called chemotaxis. That's the movement of leukocytes in response to chemical attractants that are being secreted. All right, there are 5 leukocyte types divided into 2 classes. And these classes are going to be based on whether or not you can see secretory vesicles with a light microscope. So you have the granulocytes and you have the agranulocytes. The granulocytes have these visible granules when you look at them under a microscope, and this is going to include neutrophils, eosinophils, and basophils. Then you have the agranulocytes, and these guys also have secretory vesicles, okay? But they're just really, really small, and so you can't see them with a light microscope. And this is going to include your lymphocytes and your monocytes. Okay, here's the granulocytes. You can see the neutrophil, the eosinophil, the basophil. You can see prominent granules, prominent all throughout the cytoplasm. Compare that to the agranulocytes, your lymphocytes and your monocytes. Lymphocytes, you really cannot see granules. This big dark purple spot in the center is actually the nucleus of this cell. And monocytes, you really can't appreciate granules, at least not to the extent you can with the granulocytes, and that's why they are named the way they're named. All right, let's start off by talking about neutrophils. You may also see these called PMNs. The reason for that is PMN stands for polymorphonuclear, and these guys are polymorphonuclear leukocytes, and the reason they get that name is their nucleus kind of has these weird shapes and twists and turns. It can be kind of multi-shaped, and so that's where they get that nickname. These guys are going to be the most numerous white blood cell. They are also the ones that respond first. So what's going to happen is, for example, a pathogen is going to enter the tissue, and that's going to send off signals that attract neutrophils to that tissue. You'll see neutrophils rise dramatically in infection, especially bacterial infection. Some people have a disorder called leukocyte adhesion deficiency. And what happens there is their white blood cells, chiefly the neutrophils, can't stick to the blood vessel walls in order to squeeze out of that blood vessel. So they can't stick to the vessel wall to stop themselves so they can start squeezing out. And so what ultimately happens is you don't get delivery of the neutrophils to the area where it needs to be, where they need to be. And so you can get recurrent bacterial infections with leukocyte adhesion deficiency. All right, next up in the granulocytes, we have the eosinophils. This is a much smaller percentage of white blood cells. They make up only 1 to 4% of leukocytes. They're going to have a bilobed nucleus, so here's one lobe and here's the other lobe over here. Their cytoplasm is going to stain kind of reddish pink. Sometimes you'll see it kind of orangish, but red, orange, pink, somewhere in there. They do a lot, but I want you to think of them as being primarily the defenders against parasitic worms. All right, next you have the basophils. This is an even smaller percentage of white blood cells, again with the bilobed nucleus. You can't really see it very well here, but this is one lobe and this is the other lobe. You see those prominent granules throughout the cytoplasm. Those granules contain histamine and heparin. When histamine is released, it's going to cause the blood vessel diameter to increase, so the blood vessel gets larger. That's called vasodilation. And it's also going to make the capillaries leaky, so fluid can leak out of them. That's increased capillary permeability. And you see this classically with allergy. Heparin release is going to inhibit blood clot, and so we call it anticoagulant because it inhibits blood clotting. And we'll talk about blood clotting in detail here shortly. All right, now let's talk about the agranulocytes. We'll start with monocytes. You can see a monocyte right here with its C-shaped nucleus. These guys make up about 2 to 8% of the white blood cells in the blood. And what they do is ultimately they're going to leave the bloodstream. And in doing so, they're going to transform into these large phagocytic cells called macrophages. And those macrophages hang out in the different tissues of your body where they phagocytize bacteria, viruses, cellular debris. Okay, so they're kind of the watchmen that are engulfing pathogens and potential problems and trying to get rid of them. All right, and then finally there are the lymphocytes. This is a type of agranulocyte that resides in lymphoid organs and tissues. The lymphocytes are the second most numerous white blood cells circulating in the blood. There's about 20 to 40% of the white blood cell count is lymphocytes. They have this big, round, dark staining nucleus and kind of a sliver or a crescent-shaped cytoplasm. There's three categories of lymphocytes. You have your T lymphocytes, or your T cells, your B lymphocytes, or your B cells, and the natural killer cells. Your T cells, there's a few different kinds. Some are involved in regulation of the immune response. Others are involved in directly killing infected cells. B cells, or B lymphocytes, they grew up to become these things called plasma cells. And what plasma cells do is they produce antibodies. And then natural killer cells, they patrol the body and attack abnormal, such as cancerous cells and infected cells. And we'll talk more about all of these guys in detail, as well as the other white blood cells, when we get to our chapter on the immune system. All right, some terminology here. Let's start off with differential count, or differential white blood cell count, you may hear it called. What is a differential? Well, a differential count is going to measure the amount of each type of white blood cell, and then whether any of those white blood cells are immature. And that can give you an idea of whether there's an acute infection going on, chronic inflammation, things like that. A patient can be leukopenic or have leukopenia. This is a reduced number of white blood cells, and this is going to increase that patient's risk of infection. Alternatively, a patient could have leukocytosis. This is an elevated leukocyte count. Leukocytosis can be due to a lot of things. One very common reason is just the patient has an infection that they're fighting. Stress can also increase your white blood cell count. So can inflammation, because white blood cells are involved in inflammatory processes. It could also indicate a much more serious disorder, such as a malignancy like leukemia. There's neutrophilia. This is an increase in just the neutrophils. And this is going to be associated with infections, stress, or tissue necrosis. We see neutrophilia in patients who have had a recent heart attack. So what happens is the heart is damaged, its cells die, and your body needs to send somebody in, some cleaners in, to clean up those dead cells. And so it sends in neutrophils. And so you get an increase in the number of neutrophils circulating in your blood because your body is sending them in to clean up this problem. A left-shifted differential. Don't worry about why it's called a left shift. It's named for the way that lab results used to be printed. What this is there's a real increase in the number of immature neutrophils that are in the blood. And the reason for this is you probably have something big going on, such as a really bad infection, and your body is just trying to churn out neutrophils to get them to that area, and so it starts spitting out immature neutrophils. It doesn't care. It's just trying to get neutrophils into the circulation to take care of the problem. And that's, we call that a left-shifted differential when we see those immature neutrophils enter the circulation. The opposite of neutrophilia is neutropenia. This is a decreased neutrophil count. Eosinophilia, or an increase in the number of eosinophils that are circulating. This occurs during allergic reactions, parasitic infections, especially those parasitic worms, and then some autoimmune diseases as well. Monocytes. Monocytes increase in response to chronic inflammatory disorders or even tuberculosis, and they decrease in response to prolonged steroid therapy. So one thing that steroids do is, over time at least, suppress your immune system. Okay, and then you have basophils. Why would your basophils increase? Well, if you see basophilia, it's typically going to be in response to a myeloproliferative disorder. And all that a myeloproliferative disorder means is that basically something's going on in the bone marrow. An overproduction of a particular cell is happening in the bone marrow. And the problem with that is that in that overproduction, the bone marrow is churning out immature cells. Cells that are not functional, not going to function like they should. Another problem with myeloproliferative disorders is that overproduction in one particular kind of cell can kind of crowd the bone marrow and that space that the bone marrow is in. And it can interfere with the ability of the bone marrow to produce other types of cells. So you can get a decrease in other kinds of cells during a myeloproliferative disorder. Lymphocytosis, this is going to be an increase in lymphocytes. It's going to be caused by infections, particularly viral infections, but also some chronic bacterial infections. And unfortunately, you see it in some leukemias as well as multiple myeloma. Okay, what can cause decreases in lymphocytes? Well, bad things. HIV, leukemia, and sepsis. HIV, one of the cells that it targets are going to be your helper T cells in particular. So that's a type of lymphocyte. And patients with HIV, especially if they have acquired immunodeficiency syndrome or AIDS, can have very, very, very low lymphocyte counts. And then this occurs as well in sepsis. And one of the reasons that you see a decrease in lymphocytes during sepsis is because the immune system becomes dysregulated. And that's going to lead to the lymphocytes essentially destroying themselves and eating themselves. Okay, leukemia. What is leukemia? Well, it's going to be a malignancy in those cells that eventually become leukocytes. So what you get is an abnormal development and proliferation of white blood cells. So you'll get an increase in the number of the particular affected leukocyte. And a lot of times those leukocytes are not going to be as functional as they normally should. They're abnormal. You can also see a decrease in red blood cells and the megakaryocytic lines as well. Leukemia is going to result in anemia and bleeding. There's two forms. There's acute leukemia and chronic leukemia. Acute leukemia comes on quickly, that's why it's called acute. And you typically see this in children and young adults. Chronic leukemia is going to have a much slower onset, and that's why it's called chronic. You typically see chronic leukemias in middle-aged and older individuals. Okay, and finally, platelets. Remember, these guys are small membrane-enclosed cell fragments. They don't have a nucleus. They break off of that megakaryocyte in the red bone marrow. And they play an important role in blood clotting. Normal levels, it varies, but somewhere between 150,000 to 400,000 is a normal amount of circulating platelets. About a third of your platelets are going to be stored in the spleen. And what they do is they circulate for 8 to 10 days, then they get broken down and recycled. All right, here's a blood smear, and you can see these little purple fragments here are platelets. What kind of white blood cell is this? Well, if you look at the nucleus, you'll see it's got that weird twisting, kind of convoluted shape, and remember that's polymorphonuclear shape. So this is a PMN or a neutrophil. Okay, hemostasis. What is hemostasis? Well, this is the stoppage of bleeding in response to damage to a blood vessel. Okay, so before we talk about hemostasis, let's just talk a little bit about the anatomy of a blood vessel. We'll get more in detail when we get to the vessels chapter, but just the general anatomy of a blood vessel. You're going to have blood vessel walls that are composed of layers called tunics, which surround the central lumen. So the lumen is like the open part of the blood vessel where the blood passes through. Okay, let's go from inside to outside the blood vessel tunics. So the innermost tunic, the tunic that is in direct contact with the blood, is called the tunica intima. And there, endothelial cells provide a smooth surface for blood flow. Now, all an endothelial cell is a squamous epithelial cell that makes up the inside of a blood vessel. So that's why we call them endothelial cells. And again, these endothelial cells provide a smooth surface for the blood to flow, and then they release chemicals that prevent platelets from sticking to the wall. A damaged endothelial cell will release chemicals that help platelets stick to the wall. Next, you have the middle layer of the blood vessel. This is the tunica media, and it contains smooth muscle cells that can contract in order to cause narrowing of the blood vessel, or vasoconstriction. And then finally, the outermost layer of the blood vessel wall is going to be the tunica externa. And this is just basically connective tissue that kind of holds the whole structure together. All right, so this is showing the different phases of hemostasis. And you can see, let's just take a look at the blood vessel anatomy here. This is the outermost tunic. So this is tunica externa. This is the middle tunic, the one with the smooth muscle layer. So that's tunica media. And then you have the innermost layer, and this is just these flat epithelial cells that we call endothelial cells. And that's the tunica intima. Okay, so what's happening here? Well, each one of these images is showing a different phase of hemostasis. So the first phase, the one that occurs right after injury, is going to be vascular spasm. And what happens is the injury is going to damage these endothelial cells, and these endothelial cells are going to release chemicals that trigger vascular spasm in this area, and that's going to prevent or limit blood loss. Next, you're going to have the platelet plug formation. Now, when you damage these endothelial cells, they're going to have strands of collagen hanging out of them. And what happens is those platelets bind to that collagen, and then the platelets gather more platelets, which then gather more platelets through a positive feedback mechanism until the plug is formed. And then finally, you'll enter the coagulation phase. This involves the coagulation cascade, and we'll talk about this in detail here in a minute. But it's a bunch of proteins that circulate through your bloodstream that when they sense injury, they become activated, and then they activate other proteins, which then activate other proteins, and so on and so forth until fibrin is made. And then fibrin forms the blood clot. Okay, so vascular spasm, we said, is the first phase in hemostasis. Remember, this is going to be blood vessel constriction or vasoconstriction. This is going to limit blood loss, and it can last from a few to many minutes, and platelets and endothelial cells are going to be the guys releasing chemicals. And what those chemicals do is they're going to stimulate even more vasoconstriction. And you'll see greater vasoconstriction with greater vessel damage. Then you've got the second phase of hemostasis, and this is the platelet plug formation. Normally, the vessel's endothelial wall is smooth and it's coated with prostacyclin. Prostacyclin is made by those endothelial cells, and it's an eicosanoid that helps repel platelets. So the platelets normally are not going to stick to endothelial cells. Now, what happens when a blood vessel is damaged? Well, those endothelial cells are going to be damaged, and collagen fibers within them are going to be exposed. Platelets are going to stick to those collagen fibers with the help of this thing called von Willebrand factor, and the endothelial cells produce von Willebrand factor. So platelets, in order to stick to collagen, have to have von Willebrand factor present. Then the platelets are going to stick together, aggregate, and close off the injury. Okay, next, those platelets are going to release their chemicals. This is called degranulation. So they're going to release serotonin and thromboxane A2. What these do is they cause prolonged vascular spasm. They're going to release ADP and thromboxane A2 to attract more platelets to the area and cause those platelets to degranulate. So you'll notice that's a positive feedback mechanism. And then they've got procoagulants that help stimulate coagulation, the final phase of hemostasis, as well as mitosis stimulating substances, and that's going to help trigger repair of the blood vessel. Thrombocytopenia, or low platelet count, is going to impair all phases of hemostasis, vascular spasm, the platelet plug formation, of course, as well as the coagulation phase, because platelets are involved in every single step. Now, the platelet plug is going to form quickly, but it's going to be prevented from getting too large by that prostacyclin that's being secreted by nearby endothelial cells that are not damaged. Okay, and then finally there is the coagulation phase. And coagulation means blood clotting. So when you hear someone referring to a blood clotting problem, coagulation disorder, know that that's going to affect the final phase of hemostasis. So what happens here is a network of fibrin, which is an insoluble protein, forms a mesh over that damaged area. Now, fibrin is going to come from a soluble precursor protein called fibrinogen. When it forms a mesh, it's going to entrap red blood cells, white blood cells, platelets, plasma proteins, and it's going to form a clot. Okay, what's involved in coagulation? Well, clotting is going to require the presence of calcium, the presence of clotting factors, and we'll talk about those in a second, the presence of platelets, and the presence of vitamin K. Clotting factors mostly are going to be inactive enzymes. So an inactive enzyme is going to somehow become activated, which is then going to activate another inactive enzyme, which then activates another enzyme, and so on and so forth, until ultimately fibrin is made. Most of these clotting factors are produced in the liver. This is one reason why people who have liver disease have bleeding disorders. And then you need vitamin K for several of those clotting factors, in particular factors 2, 7, 9, and 10. So 4 of those clotting factors require vitamin K in order to work. Vitamin K is a coenzyme. Incidentally, warfarin, the drug warfarin, if you know anyone on that, or it's sometimes also called Coumadin, inhibits vitamin K, and that's going to inhibit clotting factors 2, 7, 9, and 10, and that's how warfarin is used as an anticoagulant. Okay, how does the coagulation cascade start? Well, clotting is going to start with the intrinsic and extrinsic pathways, and both of those pathways converge to one common pathway. The intrinsic pathway is going to be initiated by platelets, when there is damage to the inside of a vessel wall. So the 5 steps of the intrinsic pathway, you get platelets that adhere to the vessel wall, and what they do is they release factor 12. Factor 12 converts inactive factor 11 to active factor 11. Factor 11 then changes inactive factor 9 to active factor 9. Factor 9 is going to bind with calcium, so that's where calcium comes in, as well as platelet factor 3, and it forms a complex. That complex then converts inactive factor 8 to active factor 10, active factor 8, and then active factor 8 changes inactive factor 10 to active factor 10. That's a lot of mumbo-jumbo to basically say it's a cascade. That is, one enzyme is going to convert another enzyme, which goes on to convert another enzyme, etc., etc. Or you can have the extrinsic pathway. Now this is going to be initiated by damage to the tissue outside of the vessel itself. What happens here is tissue thromboplastin is released from those damaged tissues, and it combines with factor 7 and calcium to form a complex, and then that complex converts inactive factor 10 to active factor 10. Once you get to active factor 10, you're in the common pathway. The common pathway can be activated by either the extrinsic or intrinsic pathway, as we just discussed, and it has 4 steps. Active factor 10 is going to combine with factors 2, 5, calcium, and platelet factor 3 and forms prothrombin activator. Prothrombin activator activates prothrombin to thrombin, and then thrombin converts the soluble protein fibrinogen to the insoluble protein fibrin. Factor 13 is activated in the presence of calcium, and factor 13 cross-links those fibrin molecules to produce the fibrin net, or the meshwork, the framework of the blood clot. Okay, here's all of what we just discussed laid out for you in an illustration. So we have the 2 pathways here. We have the intrinsic pathway and the extrinsic pathway, and they converge here to the common pathway. So let's say this guy here is chopping carrots and he cuts his finger. So there's going to be damage to the outside of the vessel as well as damage to the vessel itself. Well, damage to the outside of the vessel is going to trigger the extrinsic pathway. And then what you get is you get tissue factor, or factor 3, and factor 7 and calcium, and they all come together and convert inactive factor 10 to factor 10, the active form. Okay, let's pause there. What's going to happen to when the inside of the vessel is damaged by that cut? Well, the intrinsic pathway is going to be triggered. Now remember, platelets are going to release factor 12 in response to damage to the inside of a vessel, and factor 12 is going to interact with inactive factor 11 to make it active factor 11. Factor 11 then goes on and activates inactive factor 9, so now we have active factor 9. Active factor 9, along with calcium and platelet factor 3, are going to activate factor 8. Factor 8, once it's activated, can go and activate factor 10, inactive factor 10, and now you have active factor 10. Okay, everything from here down is going to be the same regardless of whether the damage is the inside of the blood vessel or outside of the blood vessel. Now, what do I expect you to know about the coagulation cascade? Well, I want you to know that it's complicated. I want you to kind of have the general sense of how this works, that one protein is going to activate another protein, which then goes on to activate another protein, and so on and so forth. I want you to know that there's a couple of different ways that you can trigger the coagulation cascade. either intrinsically, so damage to the inside of the vessel, or extrinsically, damage to the outside of the vessel. I want you to know that platelets are required to help initiate coagulation. So is calcium and vitamin K. Okay, so a lot going on with coagulation. It's a very complex process, but ultimately it's going to result in the formation of a fibrin meshwork. And that fibrin meshwork is what holds the clot together. That's the extent that I expect you to know. Okay, how do we break down a clot? I want you to focus here on fibrinolysis. So this is going to be the degradation of fibrin strands by plasmin. Now, plasmin is an active protein that's formed from the inactive precursor plasminogen, okay? The clot is going to begin to be broken down after about 2 days, within 2 days of the clot formation, and it's going to continue being broken down over a number of days. TPA, or tissue plasminogen activator, as the name suggests, interacts with plasminogen and activates it, making it plasmin. And plasmin can then go on and break down clots. Now, we use TPA therapeutically for some blood clots. For example, pulmonary embolism and ischemic stroke in the golden window. And that's generally considered to be about the first 4.5 hours after a stroke occurs. You can try to give the patient TPA and see if you can resolve that blood clot. How does the body compensate for blood loss? Well, through the sympathetic nervous system. If you lose more than 10% of your blood volume, what's going to happen is the sympathetic nervous system is going to kick in, and it's going to increase vasoconstriction, heart rate, and force of heart contraction. You're also going to see your blood being redistributed to the heart and brain. And this response is effective in maintaining blood pressure up until about 40% of blood is lost. All right, let's look at some bleeding and blood clotting disorders. So there's hemophilia. This is a group of bleeding disorders. The most common types are hemophilia A and hemophilia B. This is going to occur in an X-linked recessive pattern. A recessive means that you need 2 copies of the gene in order to express the trait. X-linked means that this gene is carried on the X chromosome. And the problem for that is when it comes to boys. Boys only need 1 copy of this gene in order to express the disease because they only have 1 X chromosome. So you'll typically see men have the full-blown disease in an X-linked recessive disease. Females are typically going to be carriers. Remember, females have 2 X chromosomes, so they have a little protection against that dysfunctional gene on 1 of the X chromosomes. Hemophilia A is a deficiency of factor 8. Hemophilia B is a deficiency of factor 9. And hemophilia C is a deficiency of factor 11. Thrombocytopenia, this is a platelet deficiency, and you could be deficient in platelets for a number of reasons, but generally speaking, it's either going to be due to an increased breakdown in platelets or decreased production of platelets. And remember, this is a problem because platelets are involved in every phase of hemostasis. And then some drugs are going to interfere with clotting. For example, aspirin. Aspirin interferes with platelet function. And warfarin, we already talked about a little bit. Warfarin interferes or inhibits vitamin K, and that's going to affect factors 2, 7, 9, and 10. Hypercoagulation. This is an increased tendency to clot. This can lead to a thrombus, and all the thrombus is a blood vessel clot. Okay, so a clot that is stuck in a blood vessel. Now, that thrombus can become dislodged. And when it becomes dislodged, it can travel through the bloodstream. And when it does that, we call it an embolus. If that embolus lodges in the pulmonary trunk or either one of the pulmonary arteries, we call that pulmonary embolism. Pulmonary embolism is a medical emergency. It can cause breathing problems and death. Hypercoagulation can also have drug-related, environmental, and genetic causes. For example, birth control pills are a big one that increases your risk for blood clots. Smoking increases your risk for blood clots. Prolonged inactivity, sitting around all day, your blood's not getting moved around like it should. You can get blood clots, especially in the legs. All right, this is a CT scan of a pulmonary embolus. So this here is the pulmonary trunk. This here is the aorta, and we're kind of looking at the heart, through a cross section here. The pulmonary trunk divides into the right and the left pulmonary arteries. And here you can see this black kind of hypodensity here. This is the clot. This is the blood clot. And so it's lodged right here at the bifurcation of the pulmonary arteries. And this can be a really big problem if it develops to the point that it occludes or blocks blood flow to the lungs. Okay, what happens to our blood as we age? Well, recall hematopoiesis. That's going to occur in the spongy bone of most bones in young children, but as we age, it's going to become restricted to just a few bones, primarily the hip bone and the sternum. Now, as we age, older red bone marrow is going to be gradually replaced with adipose tissue. And for this reason, older individuals are more likely to become anemic. They just don't have as much bone marrow to make the cells like they used to. They may also produce fewer and less active white blood cells. And that's one of the reasons why the elderly are relatively immune compromised when you compare them to at least younger people. And then we see certain types of leukemia arise more commonly in the elderly.